In this section, we're going to take a look at how cells reproduce. So kind of an introduction, cancer cells start out as normal body cells, and they'll undergo these genetic mutations from various sources, and they'll lose the ability to control tempo of their own division and cause disease. So we won't talk about it per se in this lecture, but it'll come next. So cancer therapy would seek to disrupt one or more steps in cell division. But we have to know cell division in order to kind of figure out cancer stuff. So that's part of it too, is there's so much money and funding going into cancer research these days. So if this kind of might interest you, that's a way to think about it. Okay, so another thing is that in a healthy body, we need cell division in order to grow, replace damaged cells. So if there's kind of an injury or something like that, we've got to replace those damaged cells. And also development from the embryo all the way to an adult. So development from a baby all the way to an adult. In sexually reproducing organisms, eggs and sperm result from mitosis and meiosis. But honestly, just for the cells part, we're only going to talk about mitosis. When we get to genetics, because it'll have more genetic kind of components to it, we'll come back to meiosis. But mitosis only for now. In mitosis, here's a beautiful scanning electron micrograph of two cells kind of splitting. So they're splitting. Really, in this section, we're going to talk mainly about cell division and reproduction. That's about it. A little later, we'll get into eukaryotic cell cycle and mitosis. Trying to think if that's part of this one or not. But really, we're looking at cell division. Okay, so a good essential question. Okay. How do cells divide? And how do cells divide and reproduce? That's basically it. We're going to go through the whole process of these, so you will have to know the process. Okay? And that kind of process is mitosis, so at least it might be beneficial to know that. You could also go with what is mitosis, because that's an important question here. Seems to be the first dog cameo here. She's coming over to bug us. Alright, so the ability of organisms to reproduce on their own is a key characteristic of life. So what we mean kind of, they reproduce their own kind, whereas we're going to talk about viruses later, and technically they kind of don't. They require a host cell to do their reproduction. So that's something, but one of our keys to life is that we can, organisms can reproduce their own kinds. At the cellular level, it's cell division. Okay? Reproduction at the cellular level. What it's going to do is it'll produce two daughter cells that are genetically identical to each other and the original parent cell. That's it. Okay, so these are going to be exact copies. So the step that we're in, we're talking about making exact copies. And we'll talk about why that's important. Okay, so when we do that, we actually have to duplicate the chromosomes. So we have to make copies of the chromosomes, our instructions. So remember that from the cell part stuff. So in order to do this, we've actually got to copy those instructions so we can actually make two functioning normal working cells with 2 times the instructions, 1 for each cell. Okay, and the structures that contain most of the cell's DNA. Okay, so that's the chromosome. This sorts new sets of chromosomes into resulting pairs of daughter cells. Chromosomes have the DNA, so they're doing the controlling of the cell. Think about the whole nucleus thing. That's what their whole thing is for. Living organisms reproduce by 2 different methods, one of which is asexual reproduction. This produces offspring that are identical to the original cell or organism. There are actually organisms that reproduce asexually. And it essentially does kind of what we would think of as cloning. but it's not exact copies. Well, there'll be a whole thing in genetics about that. Don't worry. Okay, but it's sort of almost like cloning because we're making exact copies of either the cell or the organism. And so this involves inheritance from all of the genes from just one parent. Sexual reproduction, on the other hand, produces offspring that are similar to the parents but show variations in those traits. So it involves inheritance of unique sets of genes from two parents. So the difference here, asexual is one parent, sexual reproduction is two parents. And cell division is used for lots of things. It can be used for reproduction of single-celled organisms, which usually gets its own name, but reproduction of single-celled organisms. It also is for growth of multicellular organisms from a fertilized egg to an adult, so growth. Growth is kind of key here. It's also for repair and replacement of cells. So when you cut yourself or you scrape yourself, you've kind of got a spot there that needs to be filled in with cells. So those repaired and replaced cells will go into that spot and will kind of grow into it. And it uses cell division to make that happen. Sperm and egg production are also undergone with cell division, but that's going to be a different one. So really the only kind of ones we'll talk about with mitosis are the first three. This is a very special form we'll talk about later. Okay, so here's asexual reproduction occurring in kind of a single cell little thing. Then we've got, this is actually a starfish and it's reproducing asexually, so it's going to undergo mitosis as well. This is a plant that we would have maybe taken off a piece of it and replanted it. That's asexual reproduction. This is clearly not asexual reproduction because there's variation. So that'd be an example of sexual reproduction producing variation in the resulting organisms. Growth and development, this occurs from mitosis. So this is what we're talking about with our cell division, exact copies. This is what we're really going to get to, is what those copies look like when that happens. So how it looks and all the stages. That's our major goal today. Okay, so when prokaryotes are little single-celled bacteria doodads that are from bacteria and archaea, those two domains, when they reproduce, it's called binary fission. Binary is like two, fission is to split in half, so really it's just dividing into two. It's not a super creative name either. Okay, but we call it binary fission when we're referring to prokaryotes. So the chromosome of a prokaryote is typically kind of circular DNA shaped. So if we've got a little bacterium and maybe it's this shaped, it'll have kind of like a circular piece of DNA in it. That's usually how we represent it. Boop. Point to. Much smaller than those of eukaryotes. So maybe bacteria, maybe, there's you a nucleus with a bunch of chromosomes in it of a eukaryote. I'm just looking for reasons to doodle today, it looks like. It's a doodly kind of day. Ooh, that might be a good Easter egg. Since it's a doodly kind of day, we'll do some parts. We're going to talk about some phases here in a little bit, and maybe we doodle those. They could just be real quick doodles. I'll show it to you. Let's do a doodly day. All right. Binary fission, back to this one, of a prokaryote occurs in 3 stages. First, we duplicate the chromosome. So we've got to make 2 copies of it, 2 copies of our instructions. Then we have continued elongation, and then we divide in 2. So it's not too hard. And when we see it, this is kind of what it looks like. So first we've got our kind of circular piece of DNA. And then we've got to duplicate that. We know that these are the instructions. So now we have 2 sets of instructions. We elongate the cell, put an exact copy of the instructions within each cell, and then we split them, and alas, we have 2 daughter cells. Okay, so not very hard. Honestly, for eukaryotes, it's not going to be a whole lot different, but it is going to require a few more steps. So here's a prokaryote in the middle of division, and you would see the chromosomes right in here, kind of a circular part in the middle. All right, now we're getting to the eukaryotic cell cycle and mitosis. And we're at the 10-minute mark, so I think this is a great time to take a break, kind of review your notes a little bit, and then come back. The eukaryotic cell cycle and mitosis, that's what we're looking at here. Eukaryotic cells are a lot more complex and larger than prokaryotic cells. Hopefully we've discovered that. If you don't kind of know that difference, let's chat about it. Come by and see me during tutoring. Let's figure that out. But eukaryotes are much more complex. They've got a lot more genes, and they store most of their genes on multiple chromosomes within the nucleus. And most of our cells will have about 46 chromosomes, though it can range from species to species. Okay. Each eukaryotic species has a characteristic number of chromosomes in each nucleus. I got ahead of myself. We as humans have 46. Okay. Eukaryotic chromosomes are composed of chromatin. This is one long DNA molecule, and there are some proteins that help maintain that chromosome structure and control the activities of the genes. For me, I like to think of chromatin, it reminds me more of like, okay, here's the cell, and here's our nucleus. It's before we ever get to anything, so it looks like spaghetti noodles, just all in the nucleus. So for me, chromatin looks like spaghetti noodles. Go with that. Okay. So to prepare for division, chromatin has to become very compact and visible with a microscope. So think about it. We've got to make copies of this stuff. So here's one, and then let's say I make more on top of it. And now if it's like a big bowl of spaghetti with 2 exact copies in it, how do you figure out the half of these things? Oh, so in order to figure out and split exactly equally the bowl of spaghetti noodles, we're going to have to make it really compact. And that's where we're going to start making some little chromosomes that we can see easier. So that'll kind of be our first stage, is that we've got to make actual chromosomes. Notice this would be that spaghetti noodly type stuff, but it's really compact in a very nice way. So that way we can separate the bowl of spaghetti exactly equally and not leave like parts of spaghetti noodles in random places, because that would be a mutation. That would be very bad. Okay, so if we start off with a chromosome, it's the DNA molecule. We're going to have to make exact copies, and we'll have to keep track of those exact copies because we need exact instructions to go into each new cell. So we're going to give them a little spot where they hook together so we can keep track of them as it goes along. Whenever we make 2 new cells, we'll put them into 2 new spaces with those exact copies. Let me know at any point if that doesn't make sense. Okay, but before a eukaryotic cell begins to divide, it duplicates all of its chromosomes, resulting in copies that we call sister chromatids. Okay, so these are exact copies, these are duplicates. 2 copies are called sister chromatids. So chromatin is spaghetti noodles. Sister chromatids are exact copies that are kind of attached, kind of for tracking. So that way we can keep track of everybody as we move along. We move through the very complicated process. The sister chromatids will be joined together at their lengths and cinched especially tightly at a narrow waist that we call the centromere. The centromere really is just the attachment point so we can keep track. We kind of hook them together. I don't know what kind of things we would use. We tie them up together. They're not leaving each other. They got to stay together. They got the buddy system working. Like a buddy system with your clone. I don't know, that freaks me out thinking I'm having to be in a buddy system with my own clone, but that's what's happening. Okay, so when a cell divides, the sister chromatids will separate from each other and are then called chromosomes. So once they separate and become independent things to do the instructions for their own cells, they will be chromosomes, and they're elevated to that independent status of chromosomes. So We may have to go through that one again, but that's the way it is. Chromosomes to start with, once they make copies, they'll be sister chromatids. When they distribute back into their daughter cells, the original cells, or their own cells that they instruct for, they'll be chromosomes again. I know. Do your best with those kind of crazy words, and we can clarify absolutely anything in class that we need to. Okay? That's what class time is for us to work through these things and make sure we got it. All right, so the cell cycle is an ordered sequence of events that extends from the time the cell is first formed, from the dividing parent cell until its own division. So it's the whole time, it's the cell lifespan almost. Okay. So we've got two phases that we'll talk about, and these are two general phases of the overall cell cycle. One is interphase. Inter means between, so this is a between phase. Okay? And it literally is that. So a between phase where we've got growth, duplication, we grow a little bit more. Okay? So there's a G1 phase where we've got growth, we increase in the amount of cytoplasm. You will need to know these. There is an S phase where we've got duplication of the chromosomes. It's called the S phase. Let's see if I can write it in here for synthesis. Oh, synthesis. So synthesis is to make. So we're making copies of the chromosomes in the S phase. G2 is really preparing for division. It's like, all right, G2, we have committed to this whole mitosis thing. We're going to do it. Let's prepare for it. Okay? The second phase is called the mitotic phase, and this is where we'll undergo mitosis and a separate thing called cytokinesis. Cytocell kinesis movement. We're going to move the cells apart. Okay, this is an important slide, so make sure you take some time on that one. Okay, here's sort of what it looks like. So if we started here, we get through the gap, the first gap, G1. Then we get into DNA synthesis, the S phase. This is where we'll duplicate chromosomes. We'll call it duplicate the DNA. Okay. Then we hit the second gap, or G2 phase, where we're really preparing for it. Then we enter a very short mitosis, where we do mitosis and then cytokinesis, the cell splitting. All right. So mitosis process through a series of stages. We've got prophase, prometaphase, metaphase, anaphase, and telophase. And you'll see this both ways, and it's okay. Sometimes you'll see prometaphase included, and sometimes you won't. The way to remember the order the old way is PMAT. Though, based on what we have, we can have kind of PPMAT, which is almost worse. So this, I think, is the perfect Easter egg. So you can either do PMAT or PPMAT, depending on how you feel. Write out the phases as we go through them. Do a little doodle. You don't necessarily have to describe it. I would in your notes. But make sure you turn it in the next class period for 5 points, 5 percentage points towards the quiz. So give me some cell division doodles. I think that'd be great. Okay. So let's keep it going. So a mitotic spindle is required to divide the chromosomes. It guides the separation of two sets of daughter chromosomes. So we've got two sets of instructions that have to be moved into each one, or each new cell. So our mitotic spindle is going to help make that happen. It has microtubules, so it's part of that cytoskeleton, and some associated proteins to make it happen. Spindle microtubules emerge from two centrosomes, so in the middle. So remember those like sister chromatids? I'm going to draw them for you. They have these little centrosomes here that attach them together. Well, the microtubules that are part of the spindle will attach to that. They're going to keep track of them and move them where they need to go. It's almost like towing it. They're going to hook on to them and then tow them to the right cells. These are some really great videos. I never can get these things to play, but I promise you we'll take a look at them a little bit later and kind of identify some of the phases. All right, so interphase looks like this. Notice we've got chromatin, and as I said before, chromatin is like a bowl of spaghetti noodles. It is incredibly difficult to separate that equally. Look at it. Can you separate it equally right now? No, you can't. Prophase, we actually will start to get these guys into a position where we might be able to move them. They're condensing. We can see our equal halves. We've got some chromosomes and sister chromatids forming. And prometaphase, there'll be a few more things coming up. So that's our starting sequence. So interphase, cytoplasmic contents doubles. We already saw that. Two centrosomes form and chromosomes duplicate. That was during our S phase. All right. Oh, this would be a good place to pause real quick, though. We're at 20 minutes. We just got through interphase. Take a little break, and then come on back. All right. Interphase might sort of look like this. It's just kind of, honestly, I'm going to say it's what your normal cell looks like. So when you look at those drawings of cells, and it's like, oh, here it is. Here's your little nucleus. Here's your endoplasmic reticulum, and your weird Golgi thing, and your little, I don't know, what, ribosomes, and what else did we have in there? Oh, we got a mitochondria. Whatever. Your normal cell that looks like this, that is interphase. Okay. There's my doodle to start with. Now we'll look at the rest of them. So prophase, the major thing that happens in prophase is in the nucleus, chromosomes become more tightly coiled and folded. We're going to make it easier to split the bowl of spaghetti noodles by tightly winding up the noodles of equal length or whatever. It's complicated. Okay, so in the cytoplasm, the mitotic spindle will begin to form and microtubules grow out of centrosomes. So that just means that we're going to get that little attachment that will help begin to kind of move those chromosomes. If you look here, we can start to see the condensing of the chromosomes. You can actually see that they're starting to form. They look a little kind of worm-like. And these are the microtubules, this kind of little burst over here that's actually going to start making its way over there. In prometaphase, the nuclear envelope will break down into fragments and disappear. We're just going to get that thing out of the way. We don't need it. We don't care about it. We're going to get it out of the way. It's just getting in our way. Microtubules will extend from the centrosomes into the nuclear region. So where we start to find those little, our little chromosomes starting to form. Some spindle microtubules will attach to kinetochores. It's just the little, like here are a little two, oh god, that's a terrible picture. Never mind, hold on. Better doodle. So where these two sister chromatids are attached together, there's a little kinetochore where they will attach at the centrosome. Other microtubules will meet those from the opposite poles. Okay, so we're starting to see, we've got a pole here, microtubules, a pole here. They're starting to shoot little microtubules out, and although we can't see it, they are attaching to the middle part of some of our chromosome squiggly lines. Okay, that's happening in prometaphase. Okay, and so you can start to see what I'm talking about. The little black dots there, those are the kinetochores. And so these things are attaching to be able to move those things equally to the opposite poles. So that's what's going to happen in metaphase, anaphase, and telophase. So the second half here. You can sort of see what the pictures look like. We got more doodles to kind of follow along here. So metaphase, the mitotic spindle is fully formed. So all those little fibers that are kind of attaching, okay, like this, those guys are fully formed. Boop. The chromosomes are lined up at the center. See, you could doodle this easy if you hadn't figured out how to doodle them yet. And then the chromosomes will line up at the cell equator. They're lined up like that. Kinetochores of sister chromatids are facing opposite poles. Those were the little black dots I showed you, so they're kind of on either side. Of where these spindles attach. do a better doodle than I did. Okay, so they're lined up. Our little things are lined up in the middle here. I am apparently having trouble doodling sideways. It's atrocious. Okay, ignore that. I'm moving forward. All right, anaphase is a pretty distinct one to see, but this is where the sister chromatids actually separate at the centromeres. Daughter chromosomes are moved to opposite poles, so those chromosomes move. And some motor proteins will help that along. But our big deal is that the spindle microtubules not attached to chromosomes will lengthen and help move everything further apart. And at the end of anaphase, we have two ends of the cell that have equal collections of chromosomes, exactly equal. They figured out that the whole process takes that whole bowl of chromatin spaghetti and still manages to get the exact copies. So that's what you can see here is they're pulling each of those chromosomes into what will eventually be a new cell for each of them. So these are being pulled each direction. Oh, my pen is hateful right now. All right, there you go. Something like that. Telophase. This is where the cell continues to elongate. You'll get that nuclear envelope starting to form around the chromosomes again. Again, kind of for protection to make sure once we've got those instructions, they are secure, they are locked away. Okay. You know, they've got security guards now. Think about your Cell City kind of analogy there. Your chromatin will uncoil again, it will relax, let its hair down, become spaghetti noodles once again. And the mitotic spindle will disappear. So all those little things that helped us move it go back to where they came from. Okay, so they'll move to either end, and we actually see this separation of the cells starting to happen. That's where we get our last little stage here called cytokinesis, and this is the actual division of cytoplasm into separate cells. So it occurs really simultaneously with telophase, so you often see telophase cytokinesis. That's kind of how that works. So in animal cells, really quickly, we've got what's called a cleavage furrow whenever we've got, it's kind of like I don't know, like those drawstring bangs. So it's almost like a string that tightens this kind of contracting ring around the two cells that breaks them in half that way. So it deepens and separates. I'll show you what that looks like. So here's kind of the cleavage furrow. And you can think of it how I think of it, like kind of like a drawstring. So it will continue to pull these drawstrings from either end, and this will get smaller and smaller until you get two separate cells. Okay, so that's essentially what cytokinesis is for animal cells. It's slightly different for plant cells because don't forget we've got the cell wall. Okay, like a fortress that's difficult to get around. But in order to separate and make new cells, we then have to create new cell walls. So we have a cell plate in the middle, and it's from vesicles, little vehicles. Little truck bring in some of that cell wall material. The plate grows outwards to reach the edges, dividing the contents into two cells. And now each cell will possess a plasma membrane and a cell wall. Real quick what that looks like. Nope, that was a video. I'll show you later. But here's sort of what it looks like for plants. Is we've got the cell wall originally, these guys divide, and then at some point we get to build the cell wall in the middle. So little vesicles, little vehicles will bring in that material. It will add up all the way up, and then eventually we'll get it to where it forms two separate cell walls for each of the daughter cells. All right, so that is where we're going to stop. I know I went really quickly to try to get us under 30 minutes or right at it, so I've moved quickly, and this is new material. So, you know, go back over it, take some time, read some of the notes, look over it. If you have any questions at all, shoot me an email. Let's figure this out, and let's make sure we get it done so we can bring it to class and understand it a little bit better.